Okay, welcome to the next challenge session. It's 3:06. I've been informed that I'm supposed to entertain you until 3:10. I haven't really planned any particular things to say, so I guess I can bloviate a bit about the big data situation. So the next three talks we're going to hear, the specific theme is around the fact that it is now possible to analyze 1 billion antibody sequences or 1 billion TCR receptors, which I think Andrew said earlier this morning is pretty insane. I remember as a grad student, I generated my first 454 data set of antibodies, which had like 1 million sequences in it. And as a lazy student, I decided that the easiest thing to do would be to submit each of these sequences 10 at a time to IMGT's website. And so the first way I tried to analyze it is by running this script over the weekend until I got an email from Patrice, which basically said, "What the hell are you doing?" So we've come a long way because it's pretty amazing now that we've seen in the tool demonstrations yesterday, and I'm sure we'll see some more later today, that the amount of kind of innovation and development and software work that's been done over the past decade has been pretty immense. And the fact that we can now think about generating billions of receptors from just a couple of individuals is pretty incredible. So with that, I think I will introduce our first speaker. It's Mikhail Shugay, who I think many of you probably know. He's been involved in a lot of repertoire related projects, such as Mixer, VDJ-DB, which I think many of you have also seen. One of the first large efforts at generating a public resource for looking at the specificity specifically of T-cell receptors. I think today he's going to be telling us about looking at motif discovery. Is that right? Great. So it's a bit of a bait and switch, he's saying. Going to be talking about large data methods for essentially summarizing these repertoires so they're easier to look at. So I guess without further ado, are you all set up? So we have, the talks are planned generally 20 minutes plus 15 minutes of discussion. But you, of course, are free to use that time as you will if you go over or not. But when we have 5 minutes left, I will give you a signal, and then I'll cut you off 5 minutes later.
Thank you.
I'm going to keep this microphone so I can run around with questions. I'll grab this for you.
Can you hear me? Yeah. So, well, this will actually be like around 22 minutes. I've measured it 3 times. So. And to start with, in this talk, I'll be mostly, as I've told previously, be focusing on the data reduction and repertoire data, AIRR-seq data decomposition. So the main goal is the main topic here will be to map a sample, a typical REP-seq sample, AIRR-seq sample of T cells, which is like million of clone types covered by 10 millions of reads. And when you get hundreds of such samples, you can get closer to 10 to the 9th into a smaller set of annotated features. And under features, I mean some summary features like statistics, for example, variable, gene usage, and insert size distribution, and also a set of specifically selected core clone types. Under core clone types, I mean, for example, the hyper-expanded T-cell clone types. And this way, the approach of this approach is, of course, from our basic testing, we can see that in order to load a large data set with 100 of such samples, even using optimized Java code, you need like 100 GB RAM. And of course, if you reduce the data, and as I'll show you later, you can reduce it more than like 100-fold sometimes, you need far less memory to load it. And also, as you have smaller data set, you can do faster computations, of course. And the other important thing is when we do comparative analysis, and when we compare clone type sets from 2 samples, the false positive number of comparisons and false positive rate grows with the sample size quadratically. So if you have less comparisons, you have less false positives. And the cons here will be, of course, what if we miss something important. For example, if there is some like expanded clonotype in a tissue that is related to autoimmune disease, however, it's really not that frequent in blood, so we just leave it out. However, we need also to see it in our comparative analysis. And so here is basically a sketch, my vision of what we get in our typical AIRR-seq sample of 10 to the 7th TCR beta reads here from the peripheral blood mononuclear cells. And around 30% of the sample, around 30% of reads will be the naive fraction. However, this fraction, it will contain almost all the sample diversity, almost all unique clone types. And it can be, as was shown previously, for example, in 2012 by Murugan and colleagues and NAS, it can be nicely decomposed into this VDJ rearrangement model that has like at most 1,000 parameters. Of course, there is also some information on selection, which we currently don't know how to parameterize. And the remaining part with most of the reads, but least of the clone types, is the expanded fraction. So you can have either monoclonal expansions, so as some specific TCR that is not homologous to anything in a data set, which is nevertheless highly expanded, like occupies 5% of the reads or more. So this is also epitope dependent, response dependence. For example, in cases of CMV infections, you can see these monoclonal expansions. You can also see prominent motifs when we have like sets of thousands of distinct TCRs. However, these 1,000 TCRs can be nicely summarized into a relatively simple position-based matrix with a selected motif. And this happens also sometimes in the response-dependent manner. For example, you have these nice epitopes of EBV and influenza. And of course, as David told previously, there can be lots of bystander activation effects, etc., some non-specific expansions. And also the things I'm not going to talk about are the presence of invariant TCRs like made T cells or NKT T cells, and of course data on the subset and phenotype of each T cell, which one can get from single cell RNA sequencing data. So I only consider conventional AIRR-seq data sets here. And the rationale for my idea for this approach is what we have observed in our recently published study in genome medicine in 2018. So we've looked at T cells that were known to be specific to certain antigens, and we looked at the difference in their frequencies. We looked at how the frequency varies between T cell sets specific to certain epitopes, and we've seen really huge variance, like 2 orders of magnitude here. The y-axis is a rearrangement frequency, and which is also nice approximation to the observed frequency in the repertoires, and the x-axis is an epitope that is recognized by a given TCR. So you can see there is a huge variance that different epitopes can be recognized by either very rare TCRs, or some of them are recognized by highly public TCRs. But the interesting thing here is that we also found out that this difference in frequency can be easily parameterized using this VDJ rearrangement model. So this is basically our approach to simulate the data. So we use the VDJ rearrangement model to generate sequences, and then we take our sequence, our TCR, and count the number of times we observe a homologous TCR in the simulated data set. And this way we get the theoretical rearrangement probabilities on x-axis and observed rearrangement frequency on y-axis. And as you can see here, there is almost a perfect correlation between them, R² equals 0.9. So this relatively simple model can actually parameterize all the difference we see between different epitope-specific T cells. And the rationale for the second part, which is the expanded clone type part, so the need to include expanded clone type into the analysis, it stems from our observation that you can see a huge difference in and the frequency of T cells specific to certain epitopes based on HLA restriction of these epitopes. And you see this for common pathogens. For example, if you have EBV and 95% of healthy adults have EBV, and you have a specific HLA allele, you will have a matched TCR that is known to recognize an EBV epitope presented by this allele in your repertoire almost certainly. So you can see a huge difference here in the frequency of T cells. The red are the ones that are found in HLA matched samples, and the black that are in HLA mismatched samples. And of course, this is far better described in this study by David and colleagues. So you even see this really cool imprint heat map, which is imprinted in HLA restricted manner. So this paper must read, and it has far more deeper insight on how this imprint is formed and what is there in the imprint. So to sum up, from this rationale, we can suggest that we can decompose our AIRR-seq data into the background, which is mostly the VDJ rearrangement model, and into a set of some specifically selected clonal expansions. And they will be HLA restricted, and this will be both ongoing response, like polyclonal and monoclonal expansions, and also the imprint of immunological memory. And we have software tools to do most of these things. For example, you can learn the VDJ arrangement model using the AIRRC software by Alexander Valzac and Thierry Moraleb. And you can also use the recently developed algorithm that was recently developed by us, AIRRC and TCR-net algorithm, to find the presence of an ongoing response, and this is mostly related to polyclonal responses. And if you want to look at immunological memory for common infections, you just use the VDJDB database and map against it. So this is just to outline the general pipeline for data reduction. You start with clonal tables, you build TCR similarity networks by connecting TCRs with homologous, for example, CDR3 amino acid sequences. You then apply this TCR-net and LS algorithm to find some specific hubs that are enriched in neighbors that have more neighbors than expected by random, either by comparing to the background control data set or to the VDJ arrangement model. And after that, you do your downstream analysis, build position weight matrices, do comparative analysis with a set of clone types. And the one thing you need to consider, as I'll tell you later why, is to add hyper-expanded clone types here. So many very interesting clonal expansions will not be captured by these two algorithms. And this is just basic statistics behind the AIRRC and TCR-net approaches. So you build your TCR similarity network. You then count the number of neighbors of your given TCR in your real data, and you compare it to the expected number of neighbors. So you take your clone type, place it into a control sample, also count its number of neighbors, and use it as a control and apply a simple Poisson test here. So the only difference between AIRRC and TCR-net, well, there are several differences in the implementation, but AIRRC uses VDJ rearrangement model and samples from VDJ rearrangement model, while TCR-net, it uses a pool of AIRRC datasets. Okay, so just to demonstrate that this approach works, several case studies. So I'm going to use, in the first part, I'm going to use a data set of the aging study by Olga Britanova, who is currently here. And there are 72 samples from healthy donors of various ages. The samples have up to 1 million of clonal types, and 3 Illumina high cyclanes, so they're quite a large data set. And I will be running comparative analysis for the full data set for the core clone types selected by TCR-net and for top 3,000 clone types selected based on their frequency. So first, this figure, this is just to show the data reduction. So here is the fraction of clone types falling into TCR-net cores, and you can see the median is 2. It's lower than 2%, I think. So you get like 50-fold, 100-fold data reduction. And interestingly, the older the individual gets, the less of its clone types are core clone types. And there are very many core chronotypes in individuals aged 0, meaning umbilical cord blood samples. So this can be mostly explained by the specific structure of VDJ rearrangements in umbilical cord blood chronotypes that don't have much non-template added bases. So if you look at the number of reads, on the other hand, to the right, you can see that for some samples, especially for elderly individuals, you can get 50% of your reads falling into this 1% of the clonotypes, which are again called as core clonotypes based on the neighborhood analysis. So you can get nice data reduction here. And as for comparative analysis, I've just ran and computed a set of distance measures implemented in the VDJ2 software. And I've ordered sample pairs by the distance in the full data set. And here it is on the x-axis, and the y-axis is the actual rank of distance measure. So the dashed line means that a complete agreement with the full data set. And as you can see, different distance metrics behave differently. For example, the D measure, which is a normalized number of unique overlapping TCR variants, there is a nice correlation between the core data set and the full data set, but not with top 3000. It's actually like more negatively correlated than positively. If you look at the F and F2 measures, with F2 a robust variant of F measures that is frequency, is a normalized frequency, so weighted by the number of reads of overlapping clonotypes, you can see a somewhat nice agreement for both cores and top 3000 clonotypes with the full data set. Again, if you look at RS, which is the correlation, but the correlation computed after you've excluded non-overlapping clone types, you can see there is better agreement for cores than for top 3000, and VJJSD, which is Jensen-Shen divergence, distance between profiles of variable and joining segment usage, is better for the top clone types, but not cores. Okay, so just here are the numbers. So as you can see, for the D measure, the number of unique clone types, you get like 85%, 85 R equals 85, which is a good approximation, but for the top 3000 clone types versus full data set, you get negative correlation. Again, for F2, you get like 0.6. So this is also a nice approximation. And for RS, it is far better to use cores than the top 3000 clone type set. As for the VJ GSD metric, the top 3000 clone types are better. However, this metric should be mostly used, I think, based on the naive fraction. So when you do data reduction and the step of building the VDJ rearrangement model from the data. And the second part is to see that not only the distances, but our capabilities of annotating the data sets are not impaired by using a smaller set of selected clone types. So this is a part of from the tutorial I'm going to give you later today. And here you can see this is an annotation of two samples and two test samples and 4 control samples from study from Emerson and colleagues in Nature Genetics. And here the gray bars represent controls, and the colored box plots, they represent the annotated samples. So one sample is called B35 positive because it has B35 allele, and other is CMV positive because it is CMV positive person. And as you can see from this plot, it's almost impossible to tell if there is any difference in the annotation profiles. Well, if you know where to look, for example, I know that here is some difference for B35 EPL, but it's absolutely impossible to see it actually. And you have, of course, lots of background. For example, most of these epitopes on this axis, so most of them are actually HIV specific epitopes. And well, you know, these persons are healthy, so there should be no HIV specific response. However, if you use only TCR-net cores and assemble clusters around them and account for HLA restriction, you get this nice clean picture. For the B35 donor, you get only one cluster, which is B35 EPL, and it's quite obvious why, because, well, you know, most of people have EBV, so this donor is presumably EBV positive. For CMV positive, you can see nice annotation for the clusters B07 restricted and A02 restricted CMV epitopes. Of course, there's not that good for A02 NLV, but if you just look at the repertoire of A02 NLV, you can see that it's too sparse to fit into a single cluster. So you get a far cleaner picture. So to sum up, it looks like you don't lose any precision. You even get rid of some noise when you apply this data reduction here in terms of annotation. And the last thing to add, why do we need to consider hyper-expanded clone types that don't form any motifs? So this is a time course from one of our old studies, hematopoietic stem cell transplant. And we know that given donor has CMV and has CMV-specific TCR re-expansion after the stem cell transplant. So we monitor a specific clone type identified by tetramer, A02 NLV here. We monitor it through the time course. And as you can see, it has a nice dynamics. So it was present before transplant, then goes up, reach stop 3, then goes up, reach stop 2. So that's really cool. It looks like some real response. But if you look at the color of the points, so black points are just all clone types, and red points are the TCR net cores. So as you can see, this clone type, it only gets some homologous neighbors, also expanded only at 37 months after the transplant. So only then it can be considered as a non-random expansion. Well, I mean, it was always there, but additional evidence only came after 37 months. While if you just look at top clone types, you can see it already at 4 months. And the reason too is the fine structure of T cells, specific T cell receptor sequences from VDJDB. Here it is yellow fever epitope, and you can see you have several nice clusters here of homologous TCRs. But on the other hand, most of the TCR sequences, they are not homologous to anything. So they don't form clusters, and that is a reality you observe for most of the epitopes. You get some nice clusters, and you get something else, and nobody knows what is it, noise or some specific forms of binding, etc. Okay, so to sum up, here are the results. So I've shown you that lots of the AIRR-seq data, especially the naive fraction of it, can be efficiently summarized using the VDGA rearrangement model, either by building it to compute summary statistics or by applying it to filter the background and give you a nice set of core clone types. And so using the core clone types, it provides you adequate accuracy for comparative analysis of samples, and also allows you to perform better, less noisy annotation of entity. I don't know what it is. Anyway, so and the last thing, this important point to use hyper-expanded clone types, which sometimes show you some specific expansions that are really important in your case, like in case of CMV-specific TCR reactivation after stem cell transplant, and they most times they are not get into any motif, so you also need to consider it. And the open question for discussion, the first is data format and dedicated algorithm software for actually doing all this pipeline and for operating with such kind of data sets. So how do we store it? Do we need to fine tune some of the distance metrics and measures like diversity measure when we operate on this kind of reduced data? The other thing is, well, how we actually compare these hyper-expanded clone types, as they don't fall into any motifs and they will be different between individuals. The question not covered here is incorporating phenotype annotations and paired chain data into the pipeline, so storing it together with reduced data. And of course, the thing, well, you know, nobody had looked at is actually doing everything mentioned before in the inverse way. So using T cells generated by the VDGA arrangement as a control, and subtracting real sample as a background to see negative selection. Okay.
No, no need.
Okay. That's all. That's all. Okay. So well, the acknowledgement section. So saying to my colleagues from Institute of Bioorganic Chemistry And thanks to my collaborators who helped me with VGGDB and comparative analysis of different AIRR-seq data benchmarking TCR-net, so David and Inkarnita, who are, I think, here, maybe. Okay, and there's a funding, and that's it. I don't know, I think it was far more than 20 minutes. No, good, perfect. So questions, discussion?
Questions? Comments?
So great talk, as usual. I just have a question that you actually ended with, which is, do we need to fine tune some of the distance and diversity measures? And what do you recommend doing in such fine tuning for data sets that you scale down somehow?
Well, I mean, the idea that follows from data reduction is that we use sequence homology to do data reduction. And one of the things with diversity, for example, diversity metrics, is that we just measure the number of unique clone types. We don't account for functional diversity. So we can have 10 completely different TCRs and 10 TCRs that differ by one base, and the diversity will be the same. So perhaps we can combine these ideas to like improve do beta metrics, and same for the overlap metric. Like we do calculate Morishita Horn index, we just use exact overlaps, but maybe we can like do this kind of data reduction, etc., to do the overlap based on one mismatch distance, something like this. This is what I mean.
Yeah, great talk. So do you think that with your approach of data reduction, which obviously works well with infectious diseases like CMV or yellow fever vaccination, do you think that in situations such as autoimmunity, where you wouldn't expect like these highly expanded clones, but maybe many low expanded, that it would still work the same way, or would you have to modify your approach for this?
Well, as for the autoimmunity studies, and I know only one successful, more or less, autoimmunity studies studied by our colleagues who were able to find actually a motive to ankylosing spondylitis. And ankylosing spondylitis is a really good autoimmune disease because it's B27 restricted, so you don't need to get much controls. And there we were looking at simply at the rearrangement probability of TCRs and the number of times it's shared in the sample. So if you get if you pull samples, and you operate with a pool sample, you use, for example, TCR-net algorithm. And in TCR-net algorithm, then there are several ways how you compute neighbors. You can compute different amino acid variants, but you can also compute neighbors on nucleotide level. So if you have a given rare rearrangement that has like 5 randomly added bases, and you have 3 different variants of it with one base interchanging, you can compute the probability of actually observing a given rearrangement several times. So you can just detect a rare clone type that was found in several different people this way, and this is the way we got the motive for this putative AS antigen in ankylosing spondylitis. So you can, this method, the TCR-net methods, it doesn't select, it not always selects highly expanded clone types. It can also select some very rare, small, abundant clone types, which are nevertheless like have several variants, and you would never expect to observe several rearrangements of, for example, this structure in like one person.
But for this, you couldn't do the data reduction. For this, you couldn't do the data reduction before the analysis, right? Because it still needs the whole data set to do this.
Yeah, right. This is also, well, how do you build an optimal pipeline to account for it? So yes,
I don't know. That's why I'm asking you.
This is a thing that should be discussed actually. So
Okay.
what we are doing currently.
Okay, thank you.
In line with, I'll ask a question.
Okay.
In line with that, it reminds me a lot of what's happening a bit in either the RNA-seq world sometimes or in metagenomics, where they're starting to focus on tools that are performing like sketches of the data essentially, which are like extremely fast and enables like a whole different kind of analysis. So this is kind of an analogous operation happening for repertoire sequencing, kind of saying. Is it fast performing these sketches?
Well, it's optimized and it runs in Java, and I can, for example, I don't know, it takes me like I think 30 minutes to collapse, to pull all these 100 samples with millions of clone types, and to then build this one mismatch tree and to find the neighbors. So, well, it's reasonably fast. It's on, I think, 40 cores, and it requires 100 GB RAM, but well, it's quite feasible. So, and afterwards, when you do cross-sample comparisons, it is far faster using the reduced data set because there are only a few clone types. And you can allow yourself, for example, to do fuzzy matches when you do cross-sample comparisons. Otherwise, you just, when you compare the full data sets, it's really, this kind of operation takes like 4 hours, for example. So, yeah, I think that that's the way to go. Well, my dream is just to operate with these TCRs like with gene expression data, like with tables, with not digital, but not highly sparse, but you know the smooth values. But I don't think it's possible.
Okay, let's thank our speaker.